Welcome Upper Sixth. So this is me going through my side, so the pathogens non-specific and specific immune response side of that test you did in October 2014. What I suggest you do is get your test out now and maybe your textbook, have it open on the right pages, the A3 post on the immune response, mark scheme and your revision guide. Just all have them to hand, and of course press pause and add notes to your paper as you go through it. I really hope this is helpful. So starting with the first one, distinguish between the structure of bacteria and viruses. Now there are 2 marks available, so you just need to do one basically. Bacteria have got this and viruses have got this, and you need to do that twice for the 2 marks. So bacteria are cells, viruses are not cells. Another word for that is acellular. Bacteria are surrounded by a cell wall. Viruses, instead of saying are not, the mark scheme says you basically have to say what it is surrounded by. So surrounded by a protein capsid, or just saying capsid is fine. Bacteria have DNA. Viruses have DNA or RNA. Remember, HIV has RNA. Bacteria have circular DNA. So that bit there in the nucleoid is a circular bit of DNA, and that little plasmid, there's circular DNA as well. By contrast, the virus has linear DNA like we do, so just a line, but you know, there it's just, that looks like a bit of RNA, doesn't it? Hope that's okay. Describe how infection with bacteria results in the production of plasma cells. Okay, so I've written the notes here, each dash means 1 mark, you can see there's 4 marks there. Then plasma cells, you can see that that's right at the end, you've got this another mark being there, so that is, we've got to get to that point. So I've drawn a little picture which is very similar to the A3 poster. It is very much, the plasma cells on the left hand side of that A3 poster, and it's all about the humoral immune response. Remember, the other one was the cell mediated response, when you've got things like the T killer cells in action. So there's your bacterial pathogen. You can see I've drawn it twice there with the cell wall, in the sense that macrophages can engulf them, digest the bacterial bit because of their lysosomes in their cytoplasm being an organelle there. The macrophage can then become an antigen presenting cell because it's leaves the antigen of the bacteria on its surface when it engulfs it. There is one T helper cell out there which has a cell surface receptor which is complementary to this antigen. Because there's probably going to be lots of these busy going around engulfing, these are quite common now, so that idea of the collision is certainly possible within 4 days. That becomes activated afterwards. And yes, that's going to reproduce, but now we're just more interested in what its job is in terms of the B cells. Now a B cell, remember, that's got an antibody on its membrane, and it needs to have this pre-activation where it too comes in contact. So there's 1 in a billion which has an antibody which is complementary shaped to the antigen. So first of all that happens, and of course remember that the T helper cells on activation releases these cytokines, which is then causes the activation, the final bits. You've got the pre-activation and activation. The B cells divide by mitosis and you get a lot of B effector cells. These then differentiate into plasma cells. Remember, these have lots of rough endoplasmic reticulum because antibodies, these little Y-shaped things, are made of protein. So there you have it. The one last thing I wanted to say was that the idea that the B cell can become an antigen presenting cell to itself as well. So here we go, macrophages become antigen presenting cell after phagocytosis, mark there. B cells become antigen presenting cells, T helper cell activated by antigen on macrophage. There you go, there's that last bit, releases cytokines, and B cells become a B effector, then plasma cells, boom. I hope that's helped. So next question, organ transplant, being given immunosuppressants, and it means you don't have cytokines. What effect? Without cytokines, basic overview, you say the immune system is weaker. They may not recover from infections. They may get opportunistic infections. So basically very similar to what happens in HIV when you get the T helper cell numbers going down, you don't get the cytokines. So really those 3 marks there would be the same as what happens with HIV as well. Why cytokines released from T helper cells Effects: no B cells, no plasma cells, no antibodies. And also remember that, there is that mark for, no T killer cells as well. That cell-mediated response wouldn't happen as well. So I hope that's helpful. So that was 3 marks. So you can get your 3 marks just from writing those things. So next question is on non-specific response. And I was just looking at your revision guide actually. So here it is, just page 40, infection and the non-specific immune response. Now you've got these barriers to prevent infection. And what you've got here is, of course, stomach acid, skin, gut and skin flora as mentioned. You've got lysozyme there, mucosal surfaces, e.g. eyes, mouth and nose, produce secretions, e.g. tears, saliva and mucus. These all contain enzymes called lysozyme. This kills bacteria by damaging their cell walls. It makes the bacteria burst open lies. So we've seen that sort of same effect when it comes to antibiotics. That'd be a tricky question. Compare lysozyme with antibiotics. All the similarities are there. What you've then got, so these are barriers you talk about. You've got this non-specific immune response. You've got inflammation there involving histamines. And you've got antiviral proteins, interferons, doing a job, which have got these 3 activation things. Then you've got phagocytosis, so all non-specific. And down here, remember inside the macrophage, it's called a lysosome, and that releases enzymes. Okay, so we just call those enzymes, but we call the ones on the surface lysozymes. So it might be released from a lysosome, but we just call them enzymes. I hope that's helped. So, here we go. Enzymes release in secretions that break down cell walls of bacteria, lysozyme. Okay, not interferon, that's to do with viruses. Histamines to do with the inflammatory response. Inflammation caused by chemical released, nice, histamines. I think you guys all got that right, so that's great. This was a stinker of a question. Now, I also, in the mark scheme, or in the paper, I gave you antigens already. That's because I was trying to make it an even number. So I've drawn a little picture along the right-hand side, and definitely in the exam you can do this. So bacteria with antigen on the surface enter blood. E produce F, which binds to D and label bacteria. Now, labelling bacteria, I hope you're immediately thinking, gosh, that's got to be an antibody. Okay, so E produce F. F must be the antibody. That's a little F by that Y-shaped thing there. What produces antibodies? Plasma cells. Okay, don't say B cells, say plasma cells. They're the specific ones. Okay, label bacteria attached onto F receptors on G. Now, what is G? Actually, you can get a bit of a hint there. G engulfs labeled bacteria by forming vacuoles. H is released into the vacuoles to destroy the bacteria. Now, that is that macrophage action. So I think that one is quite easy to jump down to H. You go, right, enzymes are released into the vacuoles, which destroy the bacteria. And G is, of course, a macrophage. Okay, this bit here is tricky. Labeled bacteria attached to F receptors on the macrophage. So that's the macrophage. Now, here's a labeled bacteria now. There's the antigen, there's the antibody. And actually there are antibody receptors, which I didn't really teach about as much detail as I should have done perhaps. So there's a receptor there on the macrophage, which is complementary to the constant region of the antibody. And that's part of the reason why now that macrophage is going to definitely engulf that pathogen, as well as that chemotaxis that that attraction that we discussed in class. So kind of 2 reasons why the macrophage, you can remember the chasing macrophage on the pathogen. It all makes a lot more sense why I would go for it. I hope that's helped. So there you got all those answers there. So B lymphocytes. Didn't I say plasma cells? Let me just add plasma there. You know what I said? Always say plasma. On the mark scheme, you do get a mark for saying B lymphocytes, but why not go plasma? Had the options there. Have a look at the mark scheme for yourself. Okay. HIV causes AIDS. What does AIDS stand for? Acquired immunodeficiency syndrome. You remember that graph that I showed you saying your T helper cells go down, and then when your T helper cells go really low, that's when you get AIDS. And that's when you've got really low T helper cells, because that's the cell that infects. So then you've got named 2 types of cells that HIV enters in the immune system. So this is quite tricky. So of course T helper cells, that should be a given. Apart from I've written /CD4 lymphocytes. Now CD4 is the name of the receptor on the T helper cell. So I'm going to draw a little picture and show you that there. And that's something, again, I didn't draw attention to enough when we taught it. Macrophages. Now that's because our cells engulf the HIV. So that's just another one. I think that was quite tricky to think about that. Explain how HIV is able to enter these cells. Now this is, most people went on to talking about reverse transcriptase and everything. Really, like, the key term there is enter. So just going through a bit like endocytosis, one from AS, so a synoptic question. There's your HIV, 2 strands of RNA, protein capsid, glycoproteins, and here's a bit of detail. The glycoprotein is called the GP12 or GP120. CD4 receptor on the T helper cell. The reason why the HIV only invades T helper is because the glycoproteins are complementary to the CD4, which only the T helper have. What you have is the fusion of the virus with the membrane. Okay, so first of all you've got the complementary glycoprotein with CD4 receptor, then you've got the fusion. Boom, done. Of course, because you've mentioned about the macrophages earlier on, and it says these cells, you can just say HIV invades the macrophage by phagocytosis. Okay? So describe the sequence of events following infection of these cells by HIV that may lead to the death of the person. Now you've got 6 marks. Death by opportunistic diseases, e.g. cancer, TB, as the final mark. And there's only 1 mark on that in the mark scheme. So it's all about making all the necessary steps to get there. So let's have a look. So the HIV has invaded the cell, the T helper cell. First of all, the viral RNA becomes DNA. Boom, mark. By reverse transcriptase, incorporates into the cell's DNA with integrase. That was another thing I didn't talk about in class. So there's an enzyme that specifically helps incorporate it. So you can imagine with gene therapy that we're using, we might utilise that enzyme as well. So production of new viruses happens. And there's no detail talking about the use R ribosomes and things like that. Then all those new viruses that are being released infect further T helper cells. Okay. T killer cells destroy T helper. Now that was the role of the T killer in the cell mediated response. So an activated T killer can come along and kill this invaded T helper cells. Okay, and what's that going to do? It's going to lower the T helper cell count, lower the immune system, e.g. no cytokines, okay, and then death. I'm going to leave it there. That's the last question. I really hope that's helped.